Perfect. Thank you so much. Okay, I'm already presenting. Okay, I'll continue. So today I will talk about the R Bioconductor tools for reusable and reproducible genomic data management and analysis. So my name is Qian Liu. I am an assistant professor from Department of Biostatistics and Bioinformatics at Roswell Park Comprehensive Cancer Center. Today, first I would like to give you some background about the current status of bioinformatics data analysis. A typical data analysis includes two main parts: the pre-processing part for data cleaning and curation, and the modeling or statistical analysis part. For example, for whole genome sequencing data, we need to do some quality check, map the sequences to the reference genome, and do the variant calling. These steps are usually based on the command line tools, such as FastQC, BWA, GATK, etc. When we get a set of candidate variants from the whole genome sequencing data, we will need to annotate the variants with their functions, and then do some filtering to remove those common variants and only retain those that are specific to your study. This part is usually done using R Bioconductor or Python. Pre-processing steps involve many different tools that are having different software dependencies. So this could be time-consuming and hard to reproduce from lab to lab. In order to enhance the reproducibility for your data analysis workflows, there are many challenges. First, people work on diverse computing environments, and complex software dependencies can easily cause installation conflicts. These issues are common in the data pre-processing steps, which are mostly based on the command line tools. Workflow languages represents a solution to these challenges. They combine different bioinformatics tools to accomplish specific data analysis tasks. Workflow languages make use of software management systems such as biocontainers to solve the software installation conflicts, or the Docker to containerize all software that are related to run a specific workflow. The common workflow language, for example, has been very widely used in different projects, consortiums, and cloud computing platforms, such as the Cancer Genomics Cloud from NCI and the Kavatka from Children's Hospital of Philadelphia. With all these advantages of using workflow language, however, there are some intrinsic challenges using them. First, the workflow language is a command line-based language. It has a non-trivial learning curve for researchers, even for skilled data analysts. Second, the workflow languages mainly deal with command line tools, which are typically involved in the pre-processing steps. It has a pre-integration with the downstream statistical analysis tools, such as R and Bioconductor. So any reproducibility issues in the modeling part are not taken care of. To address these issues, in using workflow language, we have developed two Bioconductor packages, RCWL and RSWL pipelines. These two packages are designed to connect the pre-processing steps and modeling steps and represent an R Bioconductor toolchain for reproducible bioinformatics pipelines. First, we chose one of the most widely used workflow language, Common Workflow Language, and wrote a robust and scalable R Bioconductor interface for it. And based on that, we have also developed the RSWL pipelines to catalog a set of commonly used and emerging bioinformatics tools into reproducible RSWL pipelines. Here we show the usage of these two packages. The color blocks stand for the R function names. Basically, users will start with the RSWL pipelines package to search for the required tools that are previously based on command line interface or an R package, and then load and use them directly using the R programming language. One can also build their own set of tools and can contribute to the RSWL pipelines repository. The whole data analysis workflow, including both the pre-processing and data modeling steps, then can be executed within R. Very straightforward. With one example of using STAR to index a reference genome, here I show you three different ways of using it in your data analysis for comparison. The traditional way of using star in the command line interface, you star through the CWL and you star through the RCWL. In the traditional way, first we need to download and compile the software tool. Then in most cases, we will write up an on-premise bash script to define the inputs and outputs. If we work on the HPC, we will need to configure the workload management. Different challenges that can exist. The software installation conflicts. The software tools cannot be properly tracked for its source and version once installed. Inputs and outputs between tools are usually hard-coded. We need to wait for one tool to finish and manually pass the outputs and start the next tool. This requires much human intervention, which makes the whole process time-consuming and error-prone. A workflow language solves most of these issues. Basically, we write up a CWL script to specify the base command of the tool, requirements, inputs, and outputs. Here it pulls a Docker image of a specific version for that tool, which solves the software installation issues. It can also connect different tools by passing the inputs and outputs. And then we write a YAML file to assign the values to the inputs and outputs for that tool. Lastly, we need to have a CWL runner to have your job submitted. For this step, we do need to download and install that runner. As you can see, it's very useful and convenient to streamline your data analysis. However, it does involve much work and expertise. An alternative way is to use the R version of CWL. As you do every day, you need to install the R package, and then you can take advantage of the CWL functionalities and the conveniences by using regular R functions. Going further, we have pre-built more than 200 commonly used bioinformatics tools and pipelines that are ready to be queried and used directly in R through the RCWL pipelines function. First, we can search and load pre-built tools and pipelines into the R session, and then assign values to define inputs and output parameters. We can then submit the job, which are internally submitted as a CWL scripts, but you don't need to worry about the installation or CWL runner. So if you are more of an R person and want to take advantage of the workflow language and keep everything inside R for the whole project, here I recommend you to try RCWL and RCWL pipelines. RCWL also provides functionalities if you want to build your new tools and pipelines in your research area. Here are some basic functionalities to define the inputs, outputs, tools, steps, and pipelines. There are also more advanced functions to fine-tune your tool. Here I included 2 links for the raw scripts of defining star index and star solo as a reference. On our project website, rcwl.org, we have included web pages for each of the pre-built tools and pipelines, which includes a diagram of the tool, the inputs and outputs parameters, and tool steps that are included for a pipeline. It will be helpful to browse before you dive into the R packages. RCWL has also been applied successfully to cloud computing platforms, such as Anvil, Kavatica, and Cancer Genomics Cloud, based on different working environments, such as the interactive apps of RStudio and workflow methods of CWL and Vido. To summarize, here we provide an interface of CWL in our programming environment and more than 200 pre-built tools and workflows that are highly customizable. It integrates common bioinformatics tools that are either command line based or R packages, and also the validated best practice workflows. The internal implementation uses common workflow language and software management tools such as Docker and Conda to take care of any potential reproducibility issues. Lastly, our project is open source and open development. Any contribution from the community is welcome. Next, I talk about another R package, Reuse Data, an open source, open development tool for reusable and reproducible genomic data management. Let's come back to this data analytics flow. Data analysis starts with data, and different kinds of data will be involved. Firstly, and most importantly, the experiment data, such as the RNA-seq data from patients, and then are the reusable genomic data resources, such as the reference genome, gene or variant annotation files, as these data resources will be used repeatedly in different experiments and projects. Here we'll focus on how to effectively manage and reuse these data sets. Again, here I'll show you an example of the reference genome, which will be used by all the DNA and RNA sequencing data analysis. In the traditional way, we'll first download the data, do some data pre-processing, and then we save a copy of the indexed reference genome locally and use them in different projects. There can be many challenges in data management. Without an efficient strategy for local data management, these files can be disordered, hard to find, and even redundant from repeatedly downloading due to lack of tracking of meta information, such as the source URLs and versions, etc. And individual research laboratories or shared facilities may process many different experiments with a similar analysis workflow. So repeatedly processing the same data in similar ways results in substantial inefficiencies and wasting the computing resources. And also the reproducibility of analysis pipelines involving these data files is also hampered. To address these concerns, we have developed reuse data for reusable and reproducible data management. So the first aim is to improve the data reusability and reproducibility for reusable genomic data resources. And also we want to create shareable data that are ready to pass into genomic analysis workflows. We have reimagined the tool recipes in RCWL and made them as data recipes, which are centered on the data processing and reproducible generation. Multiple tools may be involved for this process. So essentially, the data recipe is a workflow recipe with additional handling of meta information for the data. Reuse data also features the shareable curated dataset, which are fully annotated and easy to share with your lab mates and other researchers. The data recipes and curated data can be easily applied in your local data analysis and cloud computing platforms. The use of reuse data is very similar to the RCWL pipelines. Install the package, and then we can search for any pre-built data recipes. We will then evaluate the recipe and generate a local copy of the same data that is fully annotated and trackable. The data will be managed with easy search and query. Multiple datasets can be grouped together for a specific task. For example, to pass to the MuTech2 software, they can be easily converted into a JSON or YAML file to work with different workflow languages. On our project website, the rcwl.org, we presented some data recipes for some commonly used data resources, such as the leftover file from Ensemble. Each data recipe comes with a separate web page showing the data curation steps, inputs and outputs, and evaluable code chunks to use the data recipe. To summarize, today I presented you 3 Bioconductor packages for reusable and reproducible genomic data management and analysis. So the RCWL pipelines and RCWL are more tool-centered recipes, while the reuse data provide more data-centered recipes. So these tools are for reproducible bioinformatics data analysis, and the reuse data is for the reusable, reproducible genomic data resources. All of these recipes are CWL-based, and they can be applied with a unified R environment. And these recipes can be easily converted with the CWL scripts with the functionalities that are in these packages. So the tool provenance and the data provenance are retained. And we have more than 200 previewed tools and workflows for the RSW and RSW pipelines, and about 20 template data recipes for reuse data for people to use as a template to build their own data recipes. And we have this link for the cloud data sharing. So with all that, I would like to thank you and happy to take any questions.